Hello everyone, welcome to Plant Biotechnology. Today we are going to see about plant expression vector system. So usually the expression vector, which would be plasmid or virus, which is designed for gene expression in host cells. This vector can be engineered, that is genetically engineered, to have regulatory sequence that can be act as an enhancer or promoter region. The goal of well-designed expression vector is the efficient production of particular product or the specific protein molecules. And also the expression of protein may be tightly controlled through this vector, and the protein is only produced in significant quantity, okay, not more than that. Only the required volume can be generated through this vector and its controlled system. And if you see the plant gene, which can be easily inserted into the different vector system. So this vector system, which usually contains promoter region, that is very important for transcription initiation. And the next one, enhancer or silencer. So this can control the entire process through enhancing or reducing in the way of silencing its expression. And third one, we have transcriptional start site or cap site. And fourth one, we have leader sequence, which would be basically untranslated region. And fifth one, we have initiation codon. So this initiation codon, which will initiate the expression. And also we have the sixth one, that is the exons. This is the coding region which will be found in the vector system. And if you see the seventh one, where we have the introns. So this introns usually non-coding sequence. And eighth one, we have stop codon. So this stop codon, which has an important role to stop its expression or regulations. And after that, ninth one, we have second untranslated region. At last, we have the poly A tile sequence. And this is the important regions, like what are the regions we have seen it in the last slide. So which has been kept here as a diagrammatic representation. Here you have the cat box and Tata box and cap site, leader sequence, ATG. This is the starting codon, and signal peptide. It's very important to target any protein to the different target sites. And also you have the tag, that is the stop codon. At last, you will have the poly A sites. And this entire regulatory regions or the sequence will be split into two. One is the regulatory sequence, so which will contains promoter, enhancer, silencer, whereas the other regions, which will be coding regions, so this region will be calling it as the gene region. Okay. So if you see the promoter is the region of DNA sequence which helps in transcription of particular protein and also initiate the binding site for RNA polymerase enzymes. And the other two important things are in regulatory sequence, cat box and Tata box. So this box, two box are the consensus sequence which will have the promoter for RNA polymerase enzymes. And also, as I said, the starting codon, that is the ATG in mRNA initiation codon, as a mRNA initiation codon, and you have the other codons like a termination codons, TAG or TAA or TGA. At last, you will have the polyadenylation site. So this site, which will represent the end of transcription mechanism. So these are the few examples of plant expression vector system which is using frequently. So if you see the plasmids, that is the expression vector system, TI plasmid of Agrobacterium tumefaciens. So this is one of the important vector system which is frequently used and applied in many transgenic plants. And these plant viruses are also be used as the expression vector. So if you see the plant vectors, so most of the vectors are derived from the viral vector system. So here we have the DNA-based viral vector system, which is exactly derived from the virus cells. The first one, which is the cauliflower mosaic viruses, and the second one, Gemini virus, and third one, Mustard virus, and fourth one, Bagimo virus, that is the RNA-based virus vector, and fifth one, tobacco mosaic virus, and sixth one, bromo mosaic virus, and seventh one, Hardy virus, and eighth one, Potex virus, and ninth one, we have Como virus. So these are the viral vectors which can be either in DNA nature or RNA nature. So these are the virus. So it's not like if you see the other vector systems, most of the vector system would be the plasmid based, whereas here you will have the viral based vectors. If you see any other microbial applications where the plasmid based vectors are artificial chromosomes based vectors, you will be finding. In plant system, the virus has been afforded to have the expressions. This is the first viral vector system, expression vector system. The first one, Agrobacterium tumefaciens. This Agrobacterium tumefaciens is a common gram-negative soil-borne bacteria which is causing a disease named called crown gall or hairy root disease. And these bacterial genes are capable to replicate along with the plant genome. So that is more important. And also there is a synthesis of special class of components named called opines. If you see again, the Agrobacterium tumefaciens, which is attracted to the wound site via chemotoxins in response to chemicals, sugar, and phenolic molecules, which is released from the damaged cells. So the components which is releasing from the plant, which have more attraction towards the bacterium, so the transfer of transgenic genes, which will be very easy and it will be more efficient than the other methods. And here we have transferred DNA. So this transferred DNA is a tumor-inducing plasmid. If you see the tumor-inducing plasmid, this gene can cause tumor or hairy root inductions, and also which can able to produce opine biosynthesis. So these opines will be utilized for the bacterial growth or the cells growth. So if you see the tumor-inducing plasmids, so this plasmids contains 3 genes. Among the 3 genes, there are 2 genes, that is the IAAM and IAAH, which encodes enzymes which together converts the tryptophan into indole 3-acetic acids, a type of axine. If those 2 genes are deleted, then shooty crown gall disease will be produced. And third one, that is the IPT, which encodes enzymes which produce zetin type cytokinin isopentenyl adenine. So this IPT deletion, which leads to the rooty crown gall disease. So if you see the first 2 important enzymes, 2 important genes, if you delete it, you'll be getting shooty crown gall disease, whereas the third gene, if you delete it, you'll be getting the rooty crown gall disease. And here we have the entire structure. So how these genes are present in the genome. So in addition to those, another locus called TML, and the deletion of this TML, which results in large tumor, very large tumor, when you're comparing with the other deletion of genes, maybe the genes from IPT or the other IAM and the other one. So if you see this TML, which has the more influencing in creating the big tumor. Besides, this tDNA also contains genes involved in opine biosynthesis, which are located near the right border of tDNA. So if you see the left border, So here we have the direct repeats and right border. We have the NOS and TML. So this is the particular gene. So this gene is responsible because the gene size of the size of gene is pretty larger comparing with the other two important genes. So this can easily produce very larger segment of tumor. So here you can able to see the IAH and IAM and TPT is the tumor releasing. So from this, the wounded plant cell releases phenolic substance usually, and also which will have the sugar molecules, which are sensed by the VIR1A gene, and so this VIR1A gene activates VIR1G, and this VIR1G induces expression of VIR1 gene of TA plasmids, and VIR1 gene will produce all VIR1 protein. So this is the kind of sequential process from VIR1A to G and G to TA plasmid, and this TA plasmid will finally induce all the genes to produce their viral protein. So if you see here, VIR1, D1 and D2 are involved in the single strand tDNA production from TA plasmids, and also it will expose to the other cells. And the single strand tDNA, which is associated with the VIR D1 and VIR D2 with VIR E2 are exported through the transfer apparatus, that is the VIR1 BG. In plant cell, this tDNA coated with VIR1 E2 and also the various plant protein influences the transfer of tDNA plus VIR1 D1, D2, VIR1 E2 complex and the interaction of tDNA to the plant nuclear DNA. So if you any part of a gene, if you want to transfer it, so there you will have this tDNA and VIR1 D1, VIR1 D2, VIR1 E3, E2 complex. So this all will try to move into the plant cell, and there it will reach a nuclear membrane, and also finally it will attach to the plant nuclear DNA molecules. And also you have the left border and right border and plasmids and the nuclear pore complex. So these are the pods you will be finding in the genomic sequence. So here we have the complete picture. So if you see the phenolic substance and sugar molecule, and from here, so this is the particular agrobacterium species, and this agrobacterium species, so which will started from the VIR1 D1, and from the VIR1 D1, that is the VIR1A, VIR1A, from VIR1A to VIR1G, and this VIR1G will try to activate all the proteins, and from there which will try to initiate all the synthesis of VIR1 genes, that is D1, D2 and VIR1 D3, E2, and this complex, this is the VIR1 D1, D2 and VIR1 E1. So all this complex which will have the agrobacterium, and this agrobacterium which will easily transfer the genes into the host cells. This is the wounded plant. So this wounded plant, so which will make the complex, this is the VIR1 E2 and VIR1 D2 and various plant proteins which will direct it to the nuclear membrane. At the end it will have the interaction, and from this interaction which will do its transcription mechanism, translation mechanism, at last the protein molecules or the production of auxins, cytokinin or dopamine components will be segregated successfully. And here we have the list of VIR1 proteins, that is the VIR1A and VIR1G and VIR1 B1, B11 and VIR1 D1, VIR1 D2 and VIR1 E2. So if you see the function wise, this VIR1A, which is the phenolic sensor and also part of two component system with the VIR1G, and along with the phosphorylation and activates the VIR1G. And if you see the VIR1G, this is the transcriptional factor which is very useful for gene expression. If you see the B1 and B11, that is B1 to B11, the components of membrane structure for tDNA transfer. If you see D1, that is the tDNA processing and modulate VIR1 D2 activity. This D2 which is make the nick in tDNA, and this tDNA will be transferred through the BILD1B transfer apparatus. At last, you will have the BILD1E2. This is very important gene which has many functions. If you see the function wise, single stranded DNA binding protein will be happening and prevents tDNA degradation by nuclease and involved in nuclear targeting and helps in passage through the nuclear pore complex to the nuclear membrane. At last also enables the interaction with the plant genomic material. And here we have some more important points to be considered if we see the BILD1 genes, that is the T8 plasmids, which contains all the genes required for tumor formation, that is in the form of BILD1, that is the BIHER genes. So these genes will be designed, so this vector system is designed for agrobacterium mediated transformation that are taken up for the native T8 plasmids. And also you will have the tDNA border sequence, BILD1 genes, and along with that, at last, this entire genome will be modified. At the end, you will have the modified tDNA regions. So this is the another important part. T8 plasmid is a group in which that you will have the nopalin and octopine. So these are the two important T8 plasmids will be had. So if you see the nopalin type based plasmid, tumor inducing plasmid, if you see that, so here you will have the tDNA and the NC and the origin of replication, tetracycline, ACC, and the BILD1 genes. So these are the things will be there. And if you see the octopine type plasmid, T8 plasmid, so here you will have the tDNA and also tetracycline and OCAC and ORI and BILD1 genes. So these are the components will be present in both nopalin type plasmid and octopine plasmid. So if you see the expansion, here you can see that BILD1 regions and nopalin catabolism and origin of replication, and then hygroquinopine catabolisms in nopalin type. If you see the octopine, here you will have the transfer gene, that is the TRA, and then OC octopine catabolism and AC for the acropine catabolisms. So this is the another important plasmid vector, that is the Agrobacterium rhizogens. So this Agrobacterium rhizogen is a soil-borne gram-negative bacteria like the Agrobacterium, and also it causes hairy root disease of many dicotyledonous plants. And this BILD1's plasmid nature of Agrobacterium rhizogens is commonly known as the RI plasmids. So this RI plasmids, which is extensively functional homology with the tumor-inducing plasmids. So this plasmids which can enable the transfer of DNA to the plant genome through mediated segment, and this will be known as the BILD1 regions. So, and also this material, this plasmid, which will produce the agrocinopan. So here we have the entire details about the Agrobacterium rhizogens. Here you can able to see the origin of replication and col OR1 and then NPT3 and the NOS terminator LB NPT and the NOS promoter NOS terminator and multiple multiple cloning sites. Sorry. So these are the few things you'll be seeing in Agrobacterium rhizogens. So if you see the other vector system, this is the viral vector system, cauliflower mosaic virus. So here we will have the cauliflower mosaic virus, which is double-stranded in DNA in nature and which infects large range of crucifers and also especially broccos and such as cabbage and the cauliflower and the oil seed, rape and mustard. So these are the plants which will be easily affected by this cauliflower mosaic virus. And for this purpose, so this virus, which has two promoters, that is the 35S and 19S in front of its genes. Furthermore, those promoters overrides plant's own regulatory system, so it can able to control the plant system itself. So it will have more expression when you're comparing with the other viral vectors. And also this cauliflower mosaic virus 35S, well known for its promoter and also used in GM crops currently, um, which are in tested, and especially this GM based crops are maize and also other selection for plant genetic engineering, and also it will be very stronger and very consecutive promoter within that cauliflower mosaic virus. And if we see some more details, initially the bacterial DNA sequence were used to probe cauliflower mosaic virus genome and its potential sites for insertion of heterolocus genes. And this 65 base pair fragments were inserted naturally occurring XHO1 site located within the gene 2, and so this gene 2 can able to delete its genome and can accommodate this foreign gene. Once this foreign gene which is inserted, which can be easily cloned in the cauliflower mosaic virus, and this can be produced dihydrofolate reductase gene from E. coli plasmids. And the expression of BIHER gene are monitored by Western blotting, that is analyzed by the Western blotting of leaf extract. And also there are so many details they have given. And genome organization of cauliflower mosaic virus, if you see the circular double-stranded DNA that is 8 kb size represented by 2 centric circle and contains 3 discontinued adjacent to replication priming sequence. And also there is a one open circle strand while 2 are negative strands. So there is a positive strand and negative strand. So this outer arrow represents the transcription from positive strand by host DNA dependent RNA polymerase. So if you see the inner arrow represents ORF, and also the movement protein MP represent movement protein, and the aphid transcriptions factor and proteins which is the putative role in DNA folding during the encapsulation. So here you have the other regions. So that is the CP for the code protein, and RT for the reverse transcriptase, and translations transactivator protein. So here you can able to see that 19S RNA, 35S RNA, and the DNA, like there are like 3 circles. So this 3 circular DNA will be having and the ATF and the MP and the TRAP and the RT, CP, and there are 1, 2, 3, 4, all the strands are available here. And next virus, that is the Gemini virus, small circular DNA virus that can replicate in plant nuclei. And this Gemini virus vectors lack code protein gene, and also which are the families which can easily infect wide range of plant crops, including maize, cotton, wheat, bean, cassava. So these are the plants usually affected by this Gemini virus. And also, if you see the vector system has been developed for in different applications like cotton plant, and also the other applications are currently in work for rose, the plant rose, and also the vector has been used in different range of studies to silence single or multiple genes, including the meristematic gene and also proliferating cell nuclear antigens. So if you see the structure of this particular Gemini virus, so here you will have the NOC and XHO, and then you have P35S and PST1 and a LIR, ASC1 and scale and the FSC, and then you have LIR, SIR, ECO1, PST1 and BAMH1 and GUS. So these are the regions will be found in the Gemini virus. So if you see the other one, so this Gemini virus basically based on the beet curly top virus has been used to generate vaccines against hepatitis A. And another example, if you see dual model in plant activation expression platform was developed in tobacco yellow dwarf virus to control the foreign gene expression. So this virus basically belongs to the genus, are transmitted via leaf hoppers, and also most of them are infects only the monocotyledon. And there are genomes are monoparasite, and comprises of both large intrinsic regions and small intrinsic regions. And complementary sense stands codes for protein C1, and also C1 is C2 has been shown only in the viral protein. If it is necessary for application, so that time it will be synthesized. And this viral stand, this codes not only for CP V2 and also which will codes for other genes which is needed for transmissions and cell movement. So that is the MP and V1. So both V1 and V2 are needed for the systematic spread. So if you want to spread the virus, so these two proteins are very essential for spreading that particular viruses among all the plants. And their replacement will therefore limit use of resulting vectors to infect the cells in culture. So if you want to target a particular plant, so this is not a right viral vector to consider for transgenic plants. So this is the another virus, Mr.A. virus. So this Mr.A. virus has been have a lot of information here like Rep-A, Rep-B, Rep, and CP code protein and MP and V1, V2 and C1, C2 and LIR and SIR. And here we have the types of Mr.A. virus. The first one will be a wheat dwarf virus and maize streak virus, and third one tobacco yellow dwarf virus. So these viruses belongs to this group are usually whitefly transmitted infect dichotomous host, and their majority of the genome is made up of two molecules of single-strand DNA. So this DNA are similar in size and also DNA A and DNA B. The intragenic region contains two molecules of DNA, therefore named as the common region, that is the CR region. And also there is another thing for cell movement and cell replications. Also there are proteins are the genes, and cell code protein, that is the code proteins are the CP, which will be the bipartite bagomovirus encoded DNA A, and this will be used for the replication and also for the systematic spread. And this protein DNA A encodes protein which will be very useful for the replications. This is the another virus, bipartite virus, bagomovirus. So this bipartite bagomovirus, which contains two types, and those two types are listed here: African cassava mosaic virus and tomato golden mosaic virus. So if you see the structural wise here, you can able to see that CR region and the AB1 and the AC2, AC1, AC2, AC3, and the AC4 and the AB1 and the AB2 are there, and replications and trap and C code protein. So you have almost all genes which are similar to the other viral vector system. So if you see this virus, the single-stranded RNA genome, which is also served as the mRNA, it encodes at least 4 proteins in 3 open reading frame. And among these 4, we have the code protein and seems to non-essentials. And if you see the other proteins, which will be very useful for manipulating and the synthesis of recombinant messenger RNA molecule into the whole plant. And this vector consists of 2 steps. One is the use of cDNA copy of viral genome, which will be going to cloning in the E. coli cells, and second one, which will be used in the in vitro transcription of recombinant viral genome. This will code the cDNA to produce the infectious RNA copies in the plant. And this is the virus, bromomosaic virus. This is the another type viral vector. This bromomosaic virus belongs to the family Bromaviridae of plant RNA viruses. This is usually a eukaryotic RNA virus. Its replication is entirely cytoplasmic, and which will be divided into 3 RNAs, that is 1, 2, 3, and packed into a separate particles. This viral replication, which is going to be depend on the well-organized interaction between a non-structural protein, that is 1A, 2A encoded, and also this will be respectively towards the RNA1 and RNA2. And the RNA3, which is which will be different from the other two one, which is the dicystronic in nature. And if you see the non-structural movement protein, so MP protein, which promotes the cell-to-cell spread encoded through the 5' capsid protein. So, and also the translational proteins, the translation also will be happen through this capsid protein gene. And again, you have the informations on RNA 1, 2, 3, and again, this can be in a different combination, and this can give rise to the cellular RNA. And here we have the entire details. If you see RNA1, RNA2, RNA3, the size of the RNA1 is the 3.2 kb, and 2 is the 2.9, and 3 is the 2.1 kb. And the entire length they have provided, and the accommodation size they have listed out, and 5', 3' all the detail they have given it in the picture. And this is the another virus, barely strip mosaic virus. The barely strip mosaic virus has a genome consisting of 3 RNAs, that is the alpha, beta, gamma, which is encapsulated in rod-shaped virions, and also this RNAs alpha and gamma encodes protein essential for replications, and also this beta, which contains 4 open reading film. This ORF beta alpha encodes the viral capsid protein. The other encodes a triple black protein, which involved in the movement of cells towards the infected plants. This boldly strip mosaic virus RNA, that is the main beta form, can be modified to express heterologous sequence. Okay, here we have the structure of RNA alpha, RNA beta, RNA gamma. So here you can able to see the 5' to 3' end and their genes and their respective size they have given it here. And also the next one, potato virus X. This potato virus X, which is a floxus and rod-shaped virus containing plus sense RNA molecule, which can be easily genetically engineered through the, and also acting as the expression vector. And this potato virus X, which consists of replicase and capsid protein genes, and also the triple gene block, which is responsible for its movement. And here we have the PVX has produced other genes as well, and they have listed out here like HVR1 and hepatitis C viruses, and other important things are here. And next one, a Como virus, that is cowpea mosaic virus. So this Como virus cowpea mosaic virus has undergone extensive development as an expressionist vector, and this size would be 30 nanometer in diameter, and 60 large and 60 small capsid proteins are within that particular virus. And we have RNA2, and also there are some important regions which can be easily attracted through the antigenic presentation and full-length protein expressions. So this will be very useful for fusion protein productions or multimeric protein production. Production also is very useful. And these are the few references I followed for preparing this presentation. And thank you all.